In addition to its testing efforts, PGC fully utilized its sequencing bioinformatics core facilities in identifying nearly complete whole genome sequences.
When you and biotechnology, she received numerous awards. Also, as researcher microbiology curriculum in the Philippines and conducted various microbiological workshops during her watch as PSM president. She led in the efforts in the founding of the Microbiology Consortium of the Philippines and became the founding chair. Let us all welcome academician Asuncion Reymundo.
Good morning, everyone. Go ahead with the screen share. Sounds, Jello.
We can hear you, ma'am.
I would like to share with you
Talk about my
experience as a scientist, my journey into the realm of microbiology, and in the process did trailblazing in biology, hoping that somehow there's a little something that might inspire you to become microbiologists, or at least pursue a career in science, technology, engineering, and mathematics, or STEM. Let me start with a quote. Most successful men have not achieved distinction by having some new talent or opportunity presented to them. They have developed the opportunity that was at hand. I would like to think that I have been successful in my career because I developed the opportunity that was presented to me. I finished a BS degree in agriculture out of necessity, as my parents told me that I have to join my sister in Los Baños, who was then working at the UP College of Agriculture. They could not afford to support me if I studied in Manila. I made the most of the opportunity and pursued a career at UPLB. During my junior year at UPLB, we had to choose a major field of study. I was reminded of my biology class in my high school years. There was only one microscope for a class of 50 and for 5 sections. So our biology teacher was so careful about the microscope we used and would not allow anyone to fiddle with it. We simply were allowed to peep through for a minute or so, and that was it. I remember being fascinated about how we are able to see what cannot be seen by the naked eye, such as the yeast cells. Consequently, I decided to major in soil microbiology during my 3rd year in college and had the opportunity to use the microscope to my heart's content. I work on a bacterium called Rhizobium, the bacterium that grows on the roots of legumes, causing nodules and providing nitrogen to the legume plant. That was the opportunity presented to me, and I made the most out of it. After graduation, I was accepted as a research instructor at UPLB. I knew that to be with UPLB, I had to pursue graduate degree. I therefore applied for an East-West Center scholarship at the University of Hawaii and pursued an MS degree. So dear students, there are lots of scholarships for science majors. So be on the lookout when you go to college or when you pursue graduate degrees. For my thesis, I worked on a bacterium causing the wilting of tomatoes. If you go to the farm, you will see this type of wilting in some of them. I studied how it survives in the soil, which then explained why after a tomato crop with wilted plants, next crop would be wilted again, even if all the wilted crops were removed. This is the bacterium causing this wilting, Ralstonia solanacearum. By now, we all know that this is due to the fact the bacterium survives in the soil, even under dry conditions. After my master's degree, I went back to UPLB and taught microbiology and plant pathology. Subsequently, I formally transferred to the new college and became part of the forerunners of the Department of Life Sciences of the College of Science and Humanities, now College of Arts and Sciences, where microbiology was established as a formal and separate field at UPLB. After a year, I got married, and a year later, left with my husband to the University of Illinois, where he pursued a PhD degree. There, I had a child, and later I also pursued my PhD under a World Bank scholarship. There, I experienced being a wife, a mother, housekeeper, and a student all at the same time. It was an existence fraught with difficulties amidst the harsh winters of the American Midwest, but that was all worth it. So students, sometimes you have to sacrifice to obtain your goals. During my PhD days, molecular biology was at a very young stage. One incident that stuck to my mind was another project I did on the root gall or tumor of tomato. There's tumor formation in the roots. It was puzzling for me to observe the gall, which kept it growing, even if I cannot isolate the organism itself. Yet the tumor keeps on growing. It was a year later that I got the answer. Molecular biologists established that the genetic material of the bacterium here, this particular genetic material called plasmids, get transferred to the genetic material of the plant. So even if the bacterium is not there, the tumor can keep on growing bigger. The wonders of science, molecular biology indeed. One has to be open-minded, appreciate new developments. I also did research on the phenomenon of bacterial chemotaxis. This phenomenon was explained how the bacterium will be attracted to the flowers of the apple and then penetrate it, causing bacterial blight, like in here. This phenomenon is called chemotaxis. There are components in the flowers of the apples that attract the bacterium to penetrate the flowers, and then this result to blighting, to this type of disease. After my husband and I finished our PhDs, we returned to UPLB. Never did we entertain staying in the US. We really wanted to make a dent as PhD holders in the Philippines. While if we stay in the US, we're just among the hundreds of PhDs. This really sounds corny, but we really wanted to serve the country. I then started research on antimicrobials, antibiotic-producing microorganisms. We isolated and studied a lot of bacterial cultures producing antibiotics studied the antimicrobial effects of certain plants and also of sponges. I also got involved in modern biotechnology, which involves recombinant DNA technology, very early in the game, that I am referred to as one of the pioneers in the field of modern biotechnology in the Philippines. This was 1980s to 1990s. To hone me in this field, I joined workshops, seminars on the recombinant DNA technology, and finally, I did a postdoctoral work for 2 years at the International Research Institute, where I was further developed on genetic engineering recombinant DNA. When I returned to UPLB, I shared what I learned and trained students on this new technology. I offered the first course on recombinant DNA, and I shared my expertise with others even beyond UPLB by offering short courses, hands-on workshops, where participants had to go through handling of DNA and cloning techniques. This was big time in 1990s. Nowadays, it's so easy to clone certain genes. However, at that time, this was trailblazing, as our trained students or participants also trained their students on the technology. So there was a multiplier effect. Together with a US collaborator, we conducted workshops in other universities, and I got an award from the Rockefeller Foundation that allowed me to go to the laboratory of my collaborator at Kansas State University for 3 months each year for 3 years. This fellowship further honed my expertise in this field of the recombinant DNA technology. Through these projects, I saw an opportunity to establish a molecular biology lab at UPLB. And through the Rockefeller Foundation, I was able to establish a molecular biology laboratory at the National Institute of Molecular Biology and Biotechnology. Again, here's an example of making the most of an opportunity presented to me. That laboratory was also our classroom with the first recombinant DNA course that I offered. My projects using this technology were on plant pathogens, as this has immediate applications in agriculture. My first project was on this bacterium causing streaking symptoms. Look at that in the leaves of rice. This is caused by the organism Xanthomonas oryzae, PV oryzicola. We study the diversity of the organism using recombinant DNA techniques. We clone a certain gene, and that we use to differentiate the different strains. So we went through the different steps of cloning in here. I don't want to go to details about this. We studied the diversity of this organism, and so again, this was opportunity that was there, and we developed a new field at UPLB. The studies on diversity of the organism were used to devise detection techniques and strategies to deploy the right variety of rice to certain areas. Another first thing that I did was to do the first polymerase chain reaction in the Philippines. This is a method of multiplying a certain gene through several cycles of denaturation, annealing, and elongation. So after several hours before, this gene will be multiplied into 1 million copies. So, and this can be done through passing the gene to different temperatures. And that's what I did during that time, was to incubate my gene through by passing it through different incubators. So I sat there in front of the incubators, transferred my tube from one incubator to another, 3 cycles, and then back and forth the whole day. And at the end of the day, I got the first polymerase chain reaction run. I did the first polymerase chain reaction run and a PCR product. Moral of the story, just go ahead and try new discoveries. Of course, nowadays, PCR is a common tool to molecular microbiology. Another project that I got involved in heavily is on bacterial wilt of bananas. You know, your banana, table banana, the Canvendes, the Lacatan banana varieties. The organism Ralstonia solanacearum can cause wilting of the plants that produce this type of bananas. And there's the other type of disease, the blackening of the fruit, the saba, you know, the variety that you use to prepare banana Q. And we wanted to study what are causing these diseases. We applied for funding from the Australian Center for International Agriculture, and we cloned again a specific DNA piece, and we used it to differentiate the isolates from banana, cooking bananas, taba, lakatan, and even tomato varieties. Through molecular techniques, we showed that the causal organism of the wilting of this banana and the hardening of the fruit of this variety was one of the same strain. This piece of information is a critical basis of an effective disease management strategy in the banana industry. Thus, the sapa variety is no longer planted inside plantations, so it cannot be a carrier of the wilt organism, which can affect the export variety Cavendish. We did a lot of basic studies on these two microorganisms, and in the process, generating basic knowledge on their biology, published articles, while allowing a number of students to graduate. Of course, there were headaches and heartaches, and they're all part of our story. When funds do not arrive on time, and my research assistants cannot receive their salaries on time, or when materials do not arrive on time, or when experiments simply wouldn't work. But the pluses more than made up for the minuses. We got nominated and won many awards and funding for our laboratories and research projects, which even helped us in our teaching functions. As I moved up the ladder of my academic career, there came a time when I was tapped into administration. I served as director for 7 years and as dean for 5 years, but I did not give up research completely. I had to multitask as a professor, administrator, and researcher. The pinnacle of recognition as a researcher and professor was when I was elected to the National Academy of Science and Technology, the highest science policy-making body of the country. It was meant, I understand, as recognition by NAST of my contribution to the body of knowledge in biology, which I am thankful for with utmost humility. At a later time in my career, I shifted from the lab to the field, although of course I did not abandon the laboratory research work. I got involved in bioremediation, the use of microorganisms and plants, which are capable of degrading, transforming pollutants into less hazardous form, or immobilizing them. This is a field in an abandoned mine in Marinduque. This is the field when we have applied bioremediation And this is the field after several years. It's now full of growing trees. I also got involved with studying the diversity of microbial population in extreme environment. So students, if you do research, go for the less traveled road. We studied the microorganisms, the hot spring of Mount Makiling at the backyard of UP Los Baños. But it was difficult to isolate the microorganisms because they require hot temperature for growth. But we were able to fish out the genes for producing cellulose, an enzyme that degrades cellulosic materials like bagasse, rice straw, and transform them to smaller materials as substrates for other products, like perhaps paper making. We all studied the microbes from Mount Mayon. One of my students was able to isolate a bacterial strain that has antibiotic and anti-cancer properties. Here's a picture of an alkaline spring in Pangasinan, and we studied also the microbial community of this populating this spring. I have to state in any forum where my research activities are discussed that my students play a major role in my career. I pay tribute to them. These are some of my more than 100 advisees, undergraduate, MS, and PhD students. They call themselves AKR babies. As I honed them towards a biology career pathway or some other undertakings, I also learned from them. As I see them now after 45 years of service at UPLB and still serving as Professor Emeritus, I smile as I learn about their various successes in their chosen endeavors. I feel a sense of elation and great satisfaction in knowing that I contributed to what they have become. Thus, dear students who have their eyes in the future, grab the opportunities presented to you and make the most out of them. A career in science is an opportunity for a bright future for you. Well, I just present a few slides about the Philippine Science Heritage Center of the National Academy of Science and Technology. NAST encourages you students to major in STEM. And of course, I pitch for biology. NAST handles the Philippine Science Heritage Center in the picture, which is the main repository of the achievements and contributions of outstanding scientists and researchers. Currently, PHSC exhibits the hologram of our living national scientists. The PHSC again. Here's a picture of national scientist Dolores Ramirez, who is an eminent geneticist. She's still around and inspiring young ones to have a career in science. These are the women national scientists who are empowered and did and achieved a lot in their lifetime. Only, of course, national scientist Ramirez, national scientist Lourdes Cruz, national scientist Claire Baltasar, and national scientist Mercedes Concepcion are still around. The rest have joined their creator. We at NAST envision the country as a progressive Philippines anchored on science. So we promote STEM to you, our younger generation, by conducting activities like this, Meet the Scientists, or Sanlahib Symposia, or Symposium for the Youth. We also encourage you to visit the different science centers for you to know how the different fields of STEM will help our country develop into a progressive state. I end with saying that if you It is the attitude at the beginning of a difficult task, which more than anything else will define its successful outcome. So have a positive attitude towards anything in your life, and you will surely succeed. Thank you, and I wish you success in all your undertakings.
Thank you.
Thank you
That's it.
very much, Academician Raimundo, for that wonderful talk. Thank you for sharing with us more than the science, but also your career path, your experiences, not only as an educator, but also as a researcher, as a mentor, and even your administrative experiences at UPLB. And thank you for sharing your inspiring story, and also thank you for your contribution to Philippine agriculture. And I agree with you. Indeed, this generation is lucky to have all these technologies available out there. And it's a matter of finding those opportunities and seizing those opportunities. Then they can do good science and using these state-of-the-art technologies to contribute to Philippine society. So again, let's thank Academician Asuncion Raimundo for as our first resource person for this session. To our audience, we will entertain your questions during the open forum, or you may use the Q&A option to submit your questions. For our Facebook Live viewers, you may write your questions in the comment box. Okay. Also, if you're sharing about this webinar in your social media, don't forget to use the hashtags. Hashtag MYS Empowered Women, hashtag Be Excited, and hashtag Filipinast. Okay. So we'll now move on to our next speaker. From microbiology and biotechnology, we will go to molecular biology. So our next speaker is a molecular biologist who is instrumental in the creation of the Philippine Genome Center, particularly in starting the PGC DNA Sequencing Corp facility. Through this technology, high throughput DNA sequencing and analysis are made available. She, along with her colleagues, were able to sequence and analyze the genomes of multiple organisms, including pathogens of shrimp and the tall and dwarf varieties of coconut. She is the executive director of the UP Philippine Genome Center, a 2004 Outstanding Young Scientist Awardee, and a 2022 Presidential Lingkod Bayan Awardee. Friends, colleagues, ladies and gentlemen, let us all welcome Dr. Cynthia Saloma.
So good morning, everyone, and thank you very much, Nas, for the opportunity to share my story with you. This is even more remarkable considering that I am presenting after Academician Asuncion Raimundo. So can you see my slides? And can you hear me? Am I coming in loud and clear? Okay.
Very loud and clear.
Thank you so much. So good morning, everyone, again. And at the outset, I would like to thank the NAST Philippines for the invitation to speak to you today as we celebrate the month of March as Women's Month. I am Dr. Cynthia Saloma, the current Executive Director of the UP Philippine Genome Center and currently also Professor at the National Institute of Molecular Biology and Biotechnology in UP Diliman. I was privileged to have been awarded the 2004 Outstanding Young Scientist Award by the NAST Philippines in the field of biophysics. So today, I would like to share with you my cell and molecular biology story and my journey as a woman scientist and university professor, with the hope that my story will inspire many young girls and women to pursue a career in science. So this is a picture of myself, official picture from the University of the Philippines website. So you'll notice that this is our sablay, and we have to wear this with pride and distinction. And this is when we were awarded, of course, the Presidential Lincoln Point Award. So I'm going to share with you today my journey as a woman in STEM, a woman in science. The story of my journey is a testament to how far we can reach if we work hard and we have the talent and the relentless drive to succeed, to make something of ourselves in the choices we make, be it in our personal as well as in our professional lives. This is also a testament of how our ability to open ourselves to opportunities that come our way will ultimately open doors for us if we take them. Here I have outlined my story starting from my elementary years, a surprise scholarship in high school as well as in college, and also in grad school. So you will notice that my story is really a series of scholarship after scholarship after scholarship. So I began my life or my story in my elementary school. I started in the I Arroyo Elementary School in Molo, Iloilo City. This is really a walking distance from our house in Molo. Eventually, I had the opportunity to take the exam at the School for Exceptional Children in Iloilo City. So it's the School for Exceptional Children where we have students with hearing, speech, or sight impairment, but who did not let these stop them from learning. I, of course, belong to the special education fast learners class. So in many ways, the mentally gifted students are also of special needs because teachers have to constantly give us a lot of stimulation in learning, be it in art or in science, in math, and as well as, of course, in experiments and even in gardening. I was in a class with 16 other elementary students who hailed from different schools in Iloilo City. And upon passing the exam, we decided to enroll in this school. So you can just imagine that this is a school with many hyperactive students. So I initially wanted to pursue a career in law, but a generous college scholarship in fisheries made me digress from becoming a lawyer to becoming a scientist. In college, I headed the school paper as editor-in-chief, as well as also the chair of the student council. I never considered the fact that I am a girl or a woman as an impediment to leading organizations. I have many classmates and friends who never considered gender as an impediment to pursuing careers that we wanted. It could be in medicine, in law, in accountancy, in teaching, in engineering, or in business. It was a given that all of us, boys and girls alike, will go to college and pursue degrees or postgraduate degrees. So my academic life, as I mentioned, is really a series of scholarship and opportunities. Getting a scholarship is indeed a very enticing opportunity because it allows the students to study full-time. I took an exam and was luckily qualified to pursue another undergraduate degree in Japan. This is really my second undergraduate degree because I finished my degree in fisheries in 3.5 years. Despite the misgivings of my father, and after a lot of tears, I was allowed to leave for Japan and study there for the next 10 years of my life. From Japanese language school, which was 1 year in Tokyo, to 4 years as an undergraduate student at Nagoya University, to 5 years as an MS, as well as a PhD student at Osaka University. And by the way, it was also in Japan where I met my husband. So I was able to study both in different cities, Tokyo for the language, Nagoya for the molecular biology, and Osaka for the medical science degree, as well as, of course, my PhD degree in physiology. And through this experience, I was able to observe the cultures of both the Philippines and Japan, and the contrast between working as an academic in Japan and here in our country. So Nagoya University was where I studied. This is the home of 6 Nobel Prize winners in physics, in chemical biology, and so on in Japan. I was the only foreign student in my undergraduate batch, and that was in 1989. So this was my school ID during my first year of college at Nagoya University, and I was one of 10 female students in a class of 40. So they call us the S31 Kurasu, and at that time my name was, family name, of course, was Palme, so it's spelled in katakana as Paromesu Shintia. So this is my birthday. And this is Heisei Gannen Yugaku, meaning to say I entered the university in the year of, the beginning year of Heisei, and that was because there was a new emperor at that time. So it's very important in my journey that we are with a proper mentor. It's very, very important that we are under the mentorship of an individual or a person who inspires us, as well as, of course, support our endeavors. In my own story, my Japanese professor had been very supportive of my career to go to graduate school even after getting married. He helped me land a Japan Society for the Promotion of Science junior fellowship for my PhD studies. As of course, he also invited my husband to be a visiting professor in our research center. And this really opened a lot of opportunities for us to collaborate on biological imaging, particularly on confocal microscopy. So my professor, myself, and I were able to write 2 papers together. And this was a sideline for me because, of course, I do a lot of imaging, but at the same time, I'm a developmental biologist. So I would like to share with you, our students, many of you, of course, come from high school. This was my first journal publication in developmental biology, and I did this as a 4th-year college student. So this was about knockout mice, which are embryonic lethal. So by the way, we know about proto-oncogenes as well as tumor suppressor genes. And for 2 proto-oncogenes, we actually hate them because they can give rise to cancer. But at the same time, if we knock out the gene, we realize that many of these oncogenes or proto-oncogenes are actually also very, very important in the early stages of embryonic development. So in this case, this NMYC gene, which is overexpressed in many types of neural cancers, as well as in lung cancer, if knocked out, would actually give rise to an embryonic lethal mutation. So this mouse at 10.5 or 11.5 days of gestation will actually die in utero. And what have been the mutations or defects in this mouse? Obviously, there was a defect in the heart development and, of course, impairment in other organs, particularly also in the lung as well as in the brain. Here, these are pictures of what I did and part of the manuscript during my undergraduate 2 years. So I did organ culture of embryonic mouse lungs. You can just imagine. This is very small, probably less than 1 millimeter in diameter. And I had to excise this from a very, very small mouse embryo, probably 7 to 8 or 9 millimeters in length. And I have to remove this and culture this in a collagen-coated filter paper. So why did we have to do explant assembly or explant culture? The reason for this is we wanted to see what would be the consequences if the mouse lived further. Because at 10.5 or 11.5 days of gestation, the mouse already dies in utero. So what I did was, before they died, I extracted or I isolated the different organs and extended them in vitro through embryonic culture or through cell culture. So you can imagine that there were impairments in limb development, as well, of course, in long branching morphogenesis in the mutant mouse lung. If you see, and if you compare this with wild type mouse or one type embryos. So I'm fairly very happy because before graduating from undergrad, I already had a publication in a very high impact journal in my discipline, which is the Journal of Development. So for example, in the mouse that I was working on, so this is a day 9.5, 9.0 mouse embryo. This is just after embryonic turning. Many of you might not know that the mouse embryo, in order to become C-shaped, actually undergoes embryonic turning. And this is the hallmark in situ hybridization of RNA expression of the NMYC genes. So you can see them on the brachial arches, in the brain, in the spinal cord, and of course, in certain areas of the body, particularly in the neural as well as in spinal regions. So if you do whole mouth immunostaining, so this is whole mouth immunostaining, and the mouse is made to be very, very clear, like through BABB, benzyl alcohol benzoate staining, it is possible for us to visualize the branches of the different kinds of neurons during embryonic development. So these are some of the cranial ganglia in the mouse, and you can see in the mutant or in the null mutant mouse, there are some impairments in terms of axon trajectories, and then the arborization of the trigeminal ganglia, which normally would give rise to the ophthalmic, maxillary, as well as the mandibular branches. But in the NMYC mutant mouse, these branches are missing. So this is a picture of my college graduation. So I was in a Husi embroidered dress, of course, with a leather jacket, because this was winter time. And these were my batchmates or my classmates in molecular biology. So we were 10, as I remember, and I was the only foreign student in the batch. None of these ladies here, these are very extremely smart students, but none of them went on to graduate school, at least in this picture. Those shown in this picture, they went on to work for industry. I think each one of them would have been an excellent scientist. But for Japan, deciding to go to grad school, if you're a female or a woman, was really very challenging. For us Filipino women, as long as we find a good scholarship, we grab the opportunity to pursue further studies locally as well as abroad. So now I will proceed to some personal stories. So prior to graduation, so that was March 25 in Japan, the graduation is always on the 25th of March every year. On March 6, I actually got married. So about two weeks before undergraduate graduation, I got married to my husband, who is a physicist. So this is Dr. Cesar Saloma. And this was our picture just before I finished my undergraduate degree. So he fetched me and joined me for my undergraduate graduation in Japan. And we went to, and of course, this one is not, and of course, he went to my undergraduate graduation in Japan, but this was during my PhD graduation, where we went to Australia, where he attended a conference on microscopy. So after undergrad, the entire laboratory in Nagoya moved to Osaka University to be involved in complex systems development, particularly in eukaryotic organism development. So for that, I had to shift and take an exam in the medical school, where I was accepted as a graduate student in medical science. And that is also the reason why I have of my background in medical science. So we were 20 students all over Japan in a special curriculum for an MS degree in medical science, where I got to do a lot of experiments, including, of course, using a cadaver for studying anatomy. My husband came visiting as a visiting scientist for 6 months. And of course, for me, because I was already married, I, of course, also went home about 3 times a year. So during spring, summer, as well as during Christmas vacation. So these are some examples of the pictures I did for bioimaging. And this one was something I did with my husband. So this is 3D reconstruction of the virus neuron-specific antibodies. This is using a confocal microscope, which I did at Osaka University. And the next picture is a picture of our paper together, where we utilize different fluorochromes in order to increase the signal-to-noise ratio of the gene expression in the mouse embryo. So this is using a different fluorochrome, another fluorochrome. And if you're going to overlay and divide by 2, you get a much better signal-to-noise ratio. So when we submitted this paper,